Today I'll be talking about the magic of life. How everything works together in bio has always fascinated me, especially the molecular biology stuff. How everything seems to be so chaotic, but everything works beautifully together. Today I'll be looking into something called the central dogma of molecular biology. Basically, it goes through the steps of protein synthesis, and it goes through three stages: transcription, mRNA processing, and then translation. First, let's go deep into the cell, into the nucleus, and find some DNA. Now, if you look at a DNA, you see that you have the thymines, guanines, adenines, cytosines, and you also see the sugar phosphate backbones. And first, transcription starts with something called an RNA polymerase number 2. And if you look, you just go smash, and you see that the RNA polymerase attaches to DNA. Hold on, let me show you the card a bit closer. Okay, so you see the quarter now inside the card, and you see the beginning of transcription. You see that the polymerase unwinds. This process is called initiation. And if you watch, the RNA polymerase moves up and continues to form the mRNA in elongation, and finally termination when it reaches the terminator sequence. And afterwards, the polymerase just detaches. Oh, the coin fell. Detaches from the DNA, and now you got your premature RNA and the original template strand of DNA. Now you see the premature mRNA is a bit different. You see the uracil instead of the thymine, and it's also single-stranded. Before it can leave the nucleus, though, it has to go through several processes of modification. So to do that, let's first zoom out from, instead of looking at the letters, let's look at different regions of the mRNA, called the introns and the exons. Introns are the parts that serves as a buffer against certain stuff like UV radiation. So it's called the intervening regions. The other ones are the exons, the parts that are expressed. Now, to cut out those introns, we use something called snRNA, which stands for small nuclear RNAs, which binds with also other proteins to form the spliceosome. And what a spliceosome does is just pretty much cuts away those intron parts. So afterwards, you can see that I can just pinch away that intron out. And so now we have a more condensed version of mRNA. You can also recombine these exons to form more different kinds of combinations. We're not done yet, though. After removing the introns, what we have to do is add these poly A tail and the 5' cap to let the mRNA stay longer, because the nucleases are out to get those guys. So now we're done with mRNA's processing, and we now are going to go to translations. So now we're moving out into the cytoplasm, a bit out from the nucleus, and we go towards a ribosome. Now this is the first step, it's the codon recognition, but before that, let's look at the parts. You see that this guy has a small and a large part, and there's also 3 different regions. Now the first region is called the A site, or the amino acetyl tRNA binding site, which is where the tRNA enters. And then the second region is called the P site, or the peptidyl tRNA site. And then the last one, the E site, or the exit site. In each round, a new tRNA enters the A site, and then polypeptide is added. Let's show you that a bit more closely. So you have, after the first codon recognition, hold on, let's get it. Okay. After that, you can just move the tRNA into the ribosome to form the peptide bond. Now you see that the amino acid chain is getting longer, and finally you go through translocation. Pretty much the mRNA moves under it, and then the tRNA exits. This goes on and on, repeats several times until after one final translocation you form this level 1 polypeptide. You can see the amino acid chain. But this isn't done yet. As you guys know, proteins need to fold, so just like that, it turns into hemoglobin for the red blood cell. Now, it's pretty amazing how everything works, from DNA to mRNA to translation to a protein. Because if something could have gone anywhere in the process. For example, let's look at the first DNA. If, for some reason, there was a UV radiation that somehow affected the exon part of the DNA, transformed it into a mutated DNA, you see the T, that's a bit mutated now, that would have messed up the entire process. Because if you look now, the mRNA is a mutated mRNA, and even the polypeptide now has a mutated amino acid, valine, instead of GLU. And finally, that causes the blood cell to turn into a sickle cell diseased red blood cell.